Good afternoon. Welcome to the NIH Data Science Sharing and Reuse Seminar. This is sponsored by the Office of Data Science Strategy. Thank you so much for joining us and happy April. Next slide, please. As a reminder, we do use Slido for question and answer, so you'll be able to log into Slido in just one moment and ask questions of our speaker at the end of his talk. Our seminars encourage researchers to share and reuse data. We started this seminar just a little over a year ago, February of last year. Our seminar provides the opportunities for researchers to share their success stories around data sharing and data reuse. And because we work across all of our 27 institutes and centers, this is also an opportunity to learn about the highlights of data science programs within each of the institutes as we go through each institute, who brings different seminar speakers at different months. The next slide is how you log on to Slido, and there's the number there. So for our seminar today, and our first doodle poll asks you to tell us where you're joining from. We'd like to hear where you're from. We just have a few housekeeping slides before we get started. The next slide is to remind you that we are hosting a New Frontiers in Findable, Accessible, Interoperable, and Reusable Data at the BioIT World Symposium. It's held on May 3rd. I believe that you can still submit your papers if you're interested. There's a link there at the bottom, and contact my colleague. His email is below, Dr. Ishwar Chandramala Swaram, if you're interested in learning more about this symposium. The next slide is just an FYI. You still have the weekend if you're interested in submitting a supplement for your NIH-funded software program to enhance your software tools for open science. This is an opportunity to expand your software program, to enhance the program, to make the software program more reusable for other communities, or to port it to different types of computing environments. The applications are due on April 12th, and the notice for the NOSI is listed there at the bottom. The next slide is just an FYI on promoting best practices for research software sharing. There's a link there. These FAQs provide best practices for sharing software, source code that are developed under NIH grants at any stage of the development. So we hope that you'll look at those best practices. Certainly we'll be interested in your feedback. The next slide is in case you're interested in our NIH Cloud Interoperability Platform program. We do have a funding announcement open right now. Letters of intent are due next week. We are inviting applicants to provide technical, administrative, and coordination support for the Cloud Interoperability Program. There's a link there to the funding announcement. I do encourage you to look at the announcement as well as to look at the program. You'll see that we are working with 5 ICs, NHLBI for Biodata Catalyst, the Common Fund for Kids First, and NLM and CBI for dbGaP, Cancer for the Cancer Research Data Commons, and NHGRI for Anvil. So bringing together these platforms into an interoperable ecosystem is our goal, and the funding announcement is for Coordination Center. Full applications will be due the 1st of June. The next slide is just an FYI. Our next seminar will be on May 13th. We will be hearing from Dr. Karthik Kramer. She will be hosted by the I Institute. She'll be talking about leveraging her data use work to understand multi-institutional cohorts and incentivizing data sharing to support discovery. So I do hope you'll join us. She is from the Martinos Center for Biomedical Imaging at Mass General Hospital, again hosted by the National Eye Institute. The next slide is my way of introducing my colleague, Dr. Hiba Awad. She's a program director in the Repair and Plasticity Center cluster at the Division of Neuroscience at the National Institutes of Neurological Disorders and Stroke. She joined NINDS just recently in November of 2021. Welcome, Dr. Awad. So glad that you're here. She manages a portfolio for basic and translational research in traumatic brain injury, Prior to joining NIH, she was a faculty at the University of Oklahoma Health Science Center, and her research focused on understanding the mechanisms that contribute to functional defects and recovery following a mild or moderate traumatic brain injury. She does hold a doctorate from pharmacology at the University of Houston, Texas, and a bachelor's degree from pharmacy from the MN University in Jordan. I'm turning it over to you, Dr. Awad. Thank you again. Pleasure to host the NINDS Institute for today's data sharing seminar.
Thank you for the introduction, and I will start sharing. All right, so I'm very happy to be here to share a brief overview of the data science activities at the National Institute of Neurological Disorders and Stroke. And our mission at the NINDS is to seek fundamental knowledge about the brain and nervous system and to use that knowledge to reduce the burden of neurological disorders. And if you visit our website, you'll see that our strategic plan is focused on investing in the future of neuroscience, and data science plays a very big role in that. So in our NINDS vision towards a modernized, integrated, fair biomedical data ecosystem, and as you know, FAIR stands for findable, accessible, interoperable, and reusable data. We are providing the tools and analytics for data curation and harmonization, and we do have an open funding announcement. We are engaging our community through workshops and requests for information, and we always make sure to include both our research communities as well as patients, caregivers, and advocates. And with regards to developing the workforce, we realize that there is a gap in biomedical and behavioral sciences with regards to expertise in data science implementation, data visualization, data management, and data analytics. And so we are funding training grants, and we hold workshops to be able to have that interdisciplinary interaction between researchers and data scientists. We're happy to share that NINDS has been hosting and continues to host trainees through the Data Scholars Program, as well as the Code It Forward Fellowship Program, which are sponsored by the ODSS. And with regards to the data ecosystem, NINDS has invested a lot of time and effort and money into establishing the common data elements, which are a very fundamental resource in order, and a tool in order to be able to standardize and harmonize the data that is being shared in a way where we're continuing to keep the research with ultimate rigor, reproducibility, and transparency. So I encourage you to visit the website for that to utilize the tools. And with regards to having a place where all of that data can be shared, we need the data infrastructure. And so NINDS supports data repositories and knowledge bases that are specific to the NINDS research community. And this slide is, it's not inclusive of all of the data repositories, and we are working on having them available on the website soon, but this is a small snapshot of the data and tissue as well as biospecimen repositories. So we encourage everyone to look them up and to utilize the available data. So there's lots of different clinical data and data types that are available for use. So we continue to have funding opportunities, and we do that either through NINDS specific funding opportunities. And I've mentioned the one that's currently open with the deadline there. So you have plenty of time to take advantage of that. And we continue to participate with the administrative supplements program through the ODSS funding announcements. And as mentioned earlier, there is one that is currently open. So for all our attendees that are watching the webinar today, we encourage you to participate, and you do not really need to wait for a funding announcement to apply for research project grants that will utilize available data in these repositories. You can do that through the parent award mechanism. And stay tuned. As you know, in 2023, we'll be implementing the data management data sharing policy. So please keep visiting the websites, and in the next few months, there will be lots of resources that will be shared with the research community. So as you know from my introduction, I'm not a data scientist, but for all of your data science questions, please contact our data science program manager, Dr. Lindsay Scott, and she will be happy to help you with anything related to data science activities at NINDS. And so Dr. Ferguson, who is our speaker today, has applied through both of those funding mechanisms, and he currently holds awards that we're going to hear about a lot today for the Pan-Neurotrauma Data Commons, as well as his work with the Top NT Multi-Center Research Award. So with that brings us to introduce Dr. Adam Ferguson. So I'm very happy to introduce our speaker. Dr. Adam Ferguson is the Director of Data Science at the Brain and Spinal Injury Center, and he's a professor at the Department of Neurological Surgery at the Weill Institute for Neurosciences at the University of California, San Francisco. He also has a joint appointment at the San Francisco Veteran Affairs Healthcare System. Dr. Ferguson has received his PhD in psychology from Texas A&M University in behavioral neuroscience. He then went on to have 2 postdoctoral fellowships in cellular and molecular neuroscience, and he received his first NIH award which was an F32 postdoctoral fellowship. And then he moved on to a faculty position at UCSF and received his first R01 award in 2010 as an early stage investigator, and has been funded by the NIH since then. He also has received awards from the VA, which he has 2 merit awards that were focused on leveraging and harnessing big data. And he also serves as a multi-PI on an interagency resource center funded by the VA called Precise TBI. He also serves as the data science co-investigator on large multi-center clinical research projects, many of which are funded by the NIH and DoD. And he is very well respected by the neurotrauma community, both nationally and internationally. He has served on study sections, working groups, and given presentations worldwide. And he is currently serving as the Vice President-elect for the National Neurotrauma Society. He's an excellent educator and mentor, and has successfully mentored trainees through their fellowships, and they have moved on to successful scientific careers. He collectively has more than 170 publications in both preclinical and clinical research, and he has developed data sharing networks for preclinical data in spinal cord injury and traumatic brain injury. Dr. Ferguson is a champion for shaping national data sharing policy for fair data, and his most recent talk at the National Academy of Science, Engineering, and Medicine was titled "Changing the Culture of Data Management and Sharing." And I look forward to hearing your talk, which is titled "Data Sharing and Machine Intelligence for Translational CNS Injury Research." So Adam, the floor is yours.
Thank you. I appreciate the invitation to be here. Let me see if I can share my slides. Okay, so today I'm going to try to give an overview of the history of data sharing in the spinal cord injury and traumatic brain injury research community, which is very closely aligned with NINDS's mission, and give a few examples of sort of wins from applying machine learning tools to shared data that we did not collect ourselves, that were collected by others in our community. I do have some disclosures. Most of it is grants. I have a few, for-profit and nonprofit collaborations. I will not be discussing any of those today. So the premise of my talk is that CNS injury is complex. and that, may seem sort of obvious. We have over 1 trillion synapses in the human brain and over 1 billion synapses in the human spinal cord. And yet, when it comes time to classify these very complex and heterogeneous injuries to what is undoubtedly one of the most complex systems in the body, we have these very simple categorization schemes like mild, moderate, and severe traumatic brain injury. And just to illustrate how silly that is, this is 6 different examples of severe traumatic brain injury in humans. These are CT images, and you can see that there's a variety of different types of pathology that we see. And so binning them into this one category of severe is not going to help us get further ahead in targeting therapeutics to the pathology that is specifically affecting an individual. We have a similar problem in spinal cord injury. Spinal cord is an integrated processing unit with its own 1 billion synapses. It has the ability to learn, and it actually controls almost every system in the body in some way. It innervates every organ. And yet, despite this complexity, where we have injuries at multiple levels affecting every organ system, we relegate ourselves to classifying spinal cord injury into these 5 simple categories, A through E, where A is the worst and E is the best. And then when you look at sort of what we've done in the preclinical literature to try to grapple with this complexity, we've produced gazillions of tiny measures of biofunction. So I guess clinically, we have these oversimplifications. On the preclinical side, we have too much complexity, I would argue. So if you look in the literature, there's many of these multi-panel figures where people are looking at things that go up or go down in response to injury and treatment. And I think I would argue that it's difficult for us to stand back and squint at this and understand the disease of CNS injury from these types of data. So what I'm going to be arguing for today is that we, as a community, come together and try to organize the data, the raw data, put it in publicly available repositories, and then make this data AI ready so that you can operate on it with machine learning tools. And one of the things that I want to highlight here is that this raises one of these important gaps that Hiba mentioned, that this makes biomedical science absolutely dependent on this poor soul here who's doing the data entry and curation, the data wrangling. And what we found in our work, and I think many people have found this problem, is that we have many brilliant people in biomedicine, but we don't have very many who are trained in this task. And so what I'm going to be doing today is highlighting a few pioneers who have done this after they got their PhDs in biomedical sciences. So I do think that it takes sort of a combination of skills to do this work. And what this really does is it sort of flips the script on CNS injury so that it's no longer just a biomedical or a biological problem, but it really becomes a big data problem. This term big data is kind of a buzzword, but its origin is in the IT consulting field. Gartner Consulting and Analytics in the early 2000s said that modern databases are insufficient for dealing with the type of data that we're creating as humans now. And really, we need to grapple with what they called the 3 V's of big data. The first V is large data volume. So that seems pretty intuitive. That's big data. In addition, high data velocity is a big data problem. So this can be very small bits of data. Think the Twitterverse, where they're moving very quickly, and it is the velocity that makes this data hard to deal with. And then there's this third V of big data known as the data variety or wide variety problem. All 3 of these are different flavors of big data problems. And I guess I would argue that what we're dealing with in neurotrauma and CNS injury is really the problem of wide data variety. We have many thousands of variables that we collect potentially in a study, and it's the integration and combination of those that define the disease. And in a way, this is really kind of a microcosm of a broader problem in biomedical research, where if you plot the number of data sets against data size, being produced by the biomedical research community, you see that there's actually relatively little organized big data. Most of the data that is collected is in this long tail of the data distribution. So these are data sets that are relatively small in size and a great many different data sets, a high number, hold on. So there's this long tail of the data distribution where we have wildly heterogeneous data of a modest size. And at a certain point, you reach the literature limit. And beyond this, you have the long tail of dark data. So this is unpublished data. And because of meta-analyses, we now have some insight into this dark data. And it turns out that this comprises somewhere between 65 and 85% of the data that is collected in biomedical research, which is shocking if you consider that worldwide humans produce about $300 billion worth of biomedical research per year. And this, of course, means that the dark data, the value of the dark data is over $200 billion per year. So one way to phrase this is we're spending an enormous amount of time and energy as biomedical researchers to collect data that then goes into a file drawer and is never seen again. And this is not, I don't think, anyone's fault. I think it's really an artifact of the way that we disseminate data in biomedicine. And, you know, it really is this concept of scientific publication where we gather enormous reams of high quality data. We develop protocols. This is where we spend most of our time as scientists. And then with all this data, we have to curate it and annotate it and clean it and wrangle it. And through sort of this random walk process, we take hundreds to thousands of pages and we winnow it down to a 10-page high impact scientific paper. And so the problem with this, of course, is that you have increasing loss of information and potential for selective reporting. So we do have a data dissemination tool in biomedicine. It's just predicated on a 17th century technology, which is the scientific paper. So this is the first scientific paper that was published in English in 1665, and it had no data in it. It was a narrative about data that was collected. And in large part, that is still what we have in the published literature. The literature doesn't really contain raw data. It contains narratives about and summaries of data. So I guess now in sort of the 2020s, it's time for us to look at a 21st century solution. So what we've been doing in the spinal cord injury and traumatic brain injury community for the past 12 years is collecting data, raw data from multiple centers and putting it into a community accessible data repository. So we had some success early on in spinal cord injury, and this was a bit of a surprise actually. My first R01 was dedicated to this concept of building a data repository for spinal cord injury. And once we got started at conferences, people would come up to me and my postdocs and say, why don't you take my data? I mean, my data is not good enough for you to have. And so we realized very quickly that there was a huge craving to give data to people who are good at using data. So to our great surprise, we ended up unwittingly having a data management problem where everyone wanted to give us their data. And we ended up assembling 60 million data points from over 4,000 individual subjects. These are rats, mice, and monkeys, and de-identified humans with spinal cord injury. And this, in the end, was 13 centers and set the foundation for this repository that we call the Vision SCI, or Spinal Cord Injury Repository. This was sort of a hack job, I have to say. We were, not anticipating being flooded by data. So with help from the Craig H. Nielsen Foundation and then eventually the Wings for Life Foundation, we professionalized this and built an open data commons for spinal cord injury. So this is now the home that hosts the Vision SCI repository. It also has a full portal that allows people to upload their own data, manage it in a private space, and then ultimately generate persistent identifiers for their data sets and publish them in a citable format. So this was pretty successful. And for that reason, I think we were asked by the VA and NIH to develop an open data commons for TBI. And now the two of these repositories are the basis for this new U24 that we have, that's the Pan Neurotrauma Data Commons. The idea is to leverage the opportunities of this nimble and very very open community of spinal cord injury, and then the much larger and more complicated field of TBI, and bring them together into a common format for managing data. And these two ecosystems are being used by a number of sort of centralized projects within federal government. One is the NINDS Translational Outcomes Program for Neurotrauma, TOP-NT project, which is a multi-center biomarker discovery project in the preclinical space for TBI. In addition, there's this interagency award that was just went live in November that is going to be developing common data elements and managing them in an active fashion for traumatic brain injury. And then finally, there's these centralized late-stage translational projects from the VA Gordon Mansfield Spinal Cord Injury Consortium. All of these projects are harnessing these pan-neurotrauma data assets. In addition, we have a strong linkage to large-scale clinical discovery projects, the TRACK TBI project, which began with an NIH U01, and the TRACK SCI project, which is funded primarily through the DoD and private foundations. Both of these are gathering human clinical data at the point of injury, or as soon after injury as we can get it, from multiple level 1 trauma centers in the US, and building huge, highly detailed data repositories. Of course, the goal of all of these things is to make data fair. And I don't think I need to explain to this group what fair means, but basically the concept is that data sets themselves should be made findable, accessible, interoperable, and reusable. And I have to say the spinal cord injury field, and I'm proud to say this, was a very early adopter of the FAIR principles. So the FAIR principle paper came out in 2016. In fall of 2016, Lynn Jakeman from NINDS hosted a workshop, a community-facing workshop at NIH to discuss what it would take for us to make a community of spinal cord injury researchers that embraces FAIR data principles. And that was a success because it helped us gather the initial requirements for developing digital technologies that would facilitate FAIR. The following year, we convened in Washington, DC with multiple agencies, some journals, as well as spinal cord injury researchers to discuss what would be the licensing that we would need to share data in a FAIR format widely and freely in an open access way. And then the year after that, we had an implementation of a digital ecosystem that would realize the goals that were raised here and here. So what we came to after all of the sort of discussions with the community is that people wanted a continuous variable ability that the PIs had control over to share data as they wish the data to be shared. So some labs said that they wanted to have a private space where they could just keep track of their own data assets, and they wanted it to be, secure and something that the PIs had control over. They could let members of their lab in and they could share data within the lab. Others said that they wanted to be able to mint DOIs for data sets to meet journal and funder requirements. These days, a lot of journals will say, where is the DOI for the data set that undergirds this paper? And so we needed to build that infrastructure as well. So we now have this system where PIs can submit their data to their own lab. They control it. No one has access to it but them. And then when they're ready, they can choose to either share the data in sort of this preprint space that's available to all verified laboratories in the Open Data Commons, or they can request a DOI and then we release the data after an editorial review process. So this is all now in place in the form of the Open Data Commons for spinal cord injury. And as of yesterday, this ecosystem had 165 data sets from 82 labs, verified laboratories. We have about 400 users who have been registered. And this comprises about 10,000 individual subjects with spinal cord injury that are being managed in the system currently. The Open Data Commons for TBI, as you can see, sort of builds on what we did with spinal cord injury. It's a smaller community currently. There are 27 labs and 61 data sets that have been uploaded. This ends up being about 4,000 individual subjects currently. And there's a paper that is in press that's describing this ecosystem. So if you're in these systems and you've logged in, and to log in, all you need to do is sign up with an institutional email account and you're in, then you can explore public data. And you click on this and you get this list of all these publicly accessible data sets. If you go into the data sets, you end up at a landing page like this. So this provides the metadata about the data set. You have the data file here that you can download, the data dictionary that you can download. We provide a data set citation. So if people want to reuse this data, this is how they cite it. They list this as the exact citation. And the data is freely available as long as people who reuse the data attribute the credit to these authors. So they have to cite this. This is like a scientific paper is the way to think about this. And so this is all now in place and it adheres to the DataCite standards. And we hope that eventually this stuff will be indexed by NIH and others as legitimate academic work products. So what I want to do for the rest of my time is now pivot from our efforts to make data fair to what one can do once data is fair. So in other words, now that you have big data in the neurotrauma community, what can you do with it? So what we do in our lab in particular is we focus very much on this concept of machine learning based data fusion. So the idea is if, for example, you're looking at grasping recovery after a stroke or a traumatic brain injury or a spinal cord injury, the question is, how well does the individual do this? Now you can quantify that in a bunch of different ways. One way might be to have a simple clinical score where you go 0, they can't move their hand. 1, they can extend their fingers. 2, they can reach out. 3, they can touch a food item. 4, they can grasp the food item. 5, they can return it to their mouths. So that might be a simple ordinal clinical style scale. Or you can have a really elegant neurophysiological assessment where you're looking at EEG and EMG and kinematics and looking at how this movement is executed. We don't take a strong claim here on one of these being more right than the other. We consider them both pieces of information that describe this movement. So what we do in our lab is we try to plot this individual based on their score on the neurophysiology variable and their score on the grasping function. So we have a 2-variable space or bivariate space. And we're mostly interested in where this individual falls in this bivariate space. And then we're interested in how they stack up against all other individuals. So here we're looking at a bivariate outcome, as it were. The same thing can actually be done for more than 2 variables. You could have 3 or 4 or 15 or 1,000,000 variables. Ultimately, what you're concerned with is how this individual falls in the multidimensional space of the disease and how they compare against other individuals. You can do this at the level of function. You can do it at the level of surrogate biomarkers and the level of tissue changes. And if you can track the same individual throughout all these data layers, it allows you to draw linkages so that you can then come to an understanding of what it means to be a member of a group in this multidimensional space of the disease. We call this the syndromic space of the disease. And in the context of clinical work, you're really then looking at what it means to be a member of a patient group. And this becomes a tool for precision medicine because you can actually say, to what extent do these patient groups overlap, or are they discernible given the totality of all the data that you have available for these individuals? So this is the question, right? This same exact concept can be applied to translation. So now you're taking the totality of all data, and the groups you're looking at are groups of species, say rats, monkeys, and humans, and you're trying to understand the overlap among these different species given the multidimensional variables that you have at your disposal. And then finally, you can do this for precision reproducibility. Here, the concept is, to what extent do these lab groups vary, and to what extent do they overlap, given the full information that you have at your disposal, given all the variables that you can pull together. And so I want to start with an example of precision reproducibility in action. So this is work that was done by Jenny Hafley when she was a postdoc in our team. She's now an assistant professor at UCSF. And she was concerned with how neurological rehab and neurotrophic therapies could actually promote recovery. And we formed a partnership with a 3-lab group, Xialing Liu, Ray Swanson and Steve Massa, who were at the San Francisco VA. They had like a program project grant for doing complex rehab therapies for traumatic brain injury. And they collected in several hundred animals, many different variables, greater than 30 outcome variables, spanning from memory and motor function to histopathology. And many of these were collected over time. So it's a fairly typical neurological disease study in the sense that there's a lot of things you're measuring. And then the question is at the end of the study, hey, what happened? Did this drug work or did it not? And the traditional way that people ask that question is they do a whole bunch of t-tests, and they might find some things that show significance, some things that don't. And they'll focus in on those things that show significance, and then those end up in the papers, right? So there'll be these multi-panel figures where they say, hey, this drug worked. You can see that these two groups are different here. But the problem, of course, is that when you have 30 outcome variables and time, this ends up being like 6,000 t-tests. And so if you run all of the t-tests or all of the ANOVAs, you end up with a plot that looks like this. This is the p-value for each t-test. And then this is the number of tests that gave you that p-value. And you can see that the vast majority of them are not even close to significant. There are maybe a few that sort of approach significance. One thing I'd point out here is that our magical p is less than 0.05 is right here. It is exactly as likely as having a p-value of 1. So there's no, if you were to throw all of these effects into a hat and pull one out, the highest probability response is that there is no significant effect. This drug did not work. So this is the type of problem we're routinely confronted with in biomedicine. There's all kinds of complexity, and the question is really what happened and what is reproducible. So rather than go down this road of trying to do multiple hypothesis correction, a Bonferroni's test, or one of the other multiple comparison correction approaches, Jenny decided to just take all the data and do an unsupervised machine learning approach where she wasn't having to make any decisions about what worked and what didn't. So she took the full set of tissue sparing measures, health measures, and functional behavioral measures. And the real question is, to what extent do these things overlap? And using a machine learning tool, can you find the overlapping chunks of variability that are highly robust across all variables? And the tool she used is known as nonlinear principal component analysis. It's a type of unsupervised manifold learning. And once she had found this overlap, she could then ask, how do individual variables relate to this overlap? So then this becomes interpretable machine learning. And what she found is that there was this cluster of variability. There was about 24% of the total variance in the data set of outcomes. And it was the relationship between memory, attention, motor function, the laterality of the lesion, and the extent of the lesion itself. And they're all moving together as an intercorrelated unit that explained the variability in outcome. So once she had this principal component, she could actually calculate for each individual subject their unique score on this principal component 1. She could do it on orthogonal principal components. After this one was detected, the machine learning tool can zoom into other clusters of variability. And she could generate this, three-dimensional projection of the syndromic space of traumatic brain injury. And so each one of these dots is an individual animal that was projected into the syndrome of TBI. And you can see quickly that those individuals with TBI were kind of over here, and the individuals who were shams were over here. So now she knows which way is up. This way is bad. So in essence, the goal is for a therapy to take individuals that are on this part of the axis and nudge them back this way. So then she can specify PC1 as her single outcome measure for a clinical study or preclinical study. And when she does this, she flips the coin once and finds that there's this effect of a neurotrophin receptor drug that actually pushes animals back towards the shams. And this part was really surprising, that physical therapy actually inhibited the effect of this neurotrophin drug. So there was this mutual inhibition of a pro-plasticity drug and a pro-plasticity therapy of physical therapy. And they were fighting with each other and canceling each other out. So this is a pretty kind of interesting finding. It was something that was in the data, but had not been noted before because they didn't have the machine learning tools that they could bring to bear on it. And to our great surprise, this was picked up in a Nature Medicine news article about how you can find optimal drugs and doses using artificial intelligence. So this is, I guess, potentially evidence of how one can harness data that's been made fair from multiple labs. And you can see how this can actually be extended into clinical care and actually perhaps have a real impact. So there are examples of this from the TRACK-TBI study. Esther Yu, who's a neuroradiologist and physicist, and Jeff Manley, who's a neurosurgeon and neuroscientist, have some examples of this with advanced analytics applied to brain imaging. And actually, they got the first ever FDA approved biomarker for traumatic brain injury through this effort. So this is kind of the end game for applying precision medicine to neurotrauma. And the TRACK-TBI team has actually had quite a bit of luck with this. As you can see, they've developed a number of biomarkers. They've been able to characterize, the neurotrauma disease as a psychological disease. They've been able to develop point of care solutions for measuring the biomarker levels. And ultimately, for those of us that are statisticians, this has led to new developments in sort of statistical analytic approaches to try to improve, the clinical practice of analytics fused with clinical data. And so all of that, I think, is like a very useful story for illustrating how fair sharing of data can actually drive discovery and ultimately potentially precision medicine. So there is a major problem, and I think Hiba alluded to this, that sort of limits our ability to do this type of work on a large scale. And that problem is that if a neurosurgeon wants to do advanced machine learning and they have a 100-hour week where they're constantly in the OR, turns out they're not going to be able to, these days, sit down and learn Python and develop new machine learning tools. They're not going to be experts at using these tools. And so that really limits the ability to implement these tools in the clinical setting. And I summarize this problem as advanced analytics are not for hobbyists. And that's a major limitation for realizing the promise of fair data and, making data sort of not only AI ready, but usable clinically. And so to address that, we've been trying to develop open source, user-friendly tools that allow people to do advanced analytics. And the ultimate goal will be to have no code environments that promote this type of stuff so that people can do advanced analytics and use these tools in clinical decision-making. But as sort of an early illustration of this, Bel Torres Espin in our team has recently published kind of user-friendly version of an R pipeline for doing those multidimensional manifold learning things that I showed earlier. In addition, we've formed some partnerships with biotech companies and digital technology companies in the Bay Area. One of these was a company that grew out of the Stanford math department, Gunnar Carlson, who's a pioneer in topological data analysis. And we were able to apply these tools, these very advanced machine learning tools, to really old data in this paper that was pioneered by Jessica Nielsen and our team. She's now an assistant professor at University of Minnesota, but this is work that she undertook when she was an F32 fellow in our group. So basically the concept here is Jessica wanted to be able to look at how different groups of animals in spinal cord injury and traumatic brain injury cluster together. And she wanted to do this really quickly and easily. And so, we have advanced tools that can do this, but her real passion was to try to understand the richness of all of the data that we've had in the past. And one of the classic studies in the spinal cord injury community is this thing known as the multi-center animal spinal cord injury study that was a multi-site NIH-funded project in the early 90s where they were trying to develop neuroprotective therapies for spinal cord injury. And they had a multi-center randomized controlled clinical trial in rats. So as part of this, they had to develop standardized outcome measures, and they came up with this thing called the Basso-Beatty-Bresnahan scale for locomotor recovery, and they had to develop, sort of standardized injury devices. So there's this thing called the NYU Mascus Impactor that was developed. And the truth is that most of the work of this project had never been published other than, hey, there's this tool for outcome assessment and there's this tool for generating injuries. And the reason is that it was unclear to the researchers what had happened once they had done all the studies. They had too many variables to manage. So this is the paper records from Mascus. And so what Jessica did is she pulled together this team of data entry and data curation folks. And over several months of very intense labor, they turned all of this paper into AI-ready digital data. And so in essence, what they're doing is they're taking stuff that was out here in the long tail of dark data and making it organized big data. Once this was done, then she could very easily run this advanced topological data analysis. So this is a network that describes the clustering of rats with spinal cord injury. This is about 330 rats that are monitored simultaneously. And with these modern no-code machine learning tools, you can click a button and this thing unfurls, and then you can paint it with a variable of interest. And what she saw is that locomotor recovery fell into these two different clusters. Some did poorly, some did well. And then she can ask, why are these different? It wasn't because of the injury. And indeed, what she found was the variable that was most predictive was blood pressure. Specifically, animals who had high blood pressure on the table had the worst outcome. So we can then go to our clinical colleagues and say, hey, in the OR, when you're doing spinal cord injury surgeries, in the trauma center, do you keep an eye on the blood pressure? And they say, yes, we try to avoid low blood pressure. And then we say, okay, but what about the fact that high blood pressure seems to yield poor outcome? They say, we don't have any standards for that. I've always kind of wondered about that, but also, I don't know that I really trust the findings that you've shown me here, because machine learning is basically a black box, and how do we know that this effect is reproducible? So when Jessica and I were confronted with this, we then got a grant from the Wings for Life Foundation and went to New Jersey and recovered all of the original data from Mascus. This had been in a storage unit for 20 years, found these old, digital technologies, and we're able to datafy all of these old data. So we now have 1,400 animals worth of spinal cord injury data from 8 centers. And because of the Open Data Commons for spinal cord injury, we've published 2 separate DOIs for all of the Mascus study. And now that we have this sort of independent cohort for trying to reproduce Jessica's original paper, we can do things like this, where we look at all of the variables for thousands of rats simultaneously. And black here reflects data that are missing, and you can just kind of get a sense of the patterns of data that are available. And then you can develop analytic tools that help you deal with the missingness. And a success of this has been demonstrated by Carlos Almeida, who's now a PhD student at University of Louisville, where he was able to take the original findings and show that the reason why we were seeing poor outcome is because we were enriched in more severe injuries in the first cohort that we have. And in fact, the relationship between blood pressure and the amount of locomotor recovery is actually very complicated depending on the type of injury you have. And if you were to fit a curve to this, you would see this inverted U-shaped dose-response function relating blood pressure in the OR to the amount of locomotor recovery. So this is in essence a successful replication of what we found with that advanced machine learning tool. It also suggests that, maybe we have something that we should look at in the clinic. Of course, rats are not human. So to do this in a clinical context requires that you have a clinical team that you can work with. And fortunately for us, we're part of this TRACK SCI team, which has every clinical service that sees spinal cord injuries at the Zuckerberg San Francisco General Hospital, the Level 1 Trauma Center for the Bay Area, and a few other sites. So from this, we're able to assemble a 2-center cohort of 131 spinal cord injury surgeries. From this, we ended up with 118 patients worth of data that we could leverage to look clinically at the relationship of blood pressure and long-term motor recovery. This resulted in a paper that came out in November, led by Abel Torres Espin and this very large group. basically what we're able to do here is look at mean arterial blood pressure in the operating room. So each little row here is an individual patient's surgery. Each pixel is a 5-minute epoch of blood pressure. You can see that we have this over time in a very rich data set. We have heart rate and other physiology. And we could then project these individuals into the multidimensional physiome space of hemodynamic measures and end up with this topological view of patient clustering. So to zoom in here, individual patients are shown as dots, and the edges are distances. So this reflects that these two individuals are similar in a multidimensional way. And so that you can then have these advanced tools that cluster these individuals in a very flexible manner and tell you these are the unique clusters that seem to exist very robustly in these data. Once you have the clusters, you can ask, where does blood pressure fall on these clusters? And you can see very quickly that there's quite diversity here. And then you can ask, how do these groups of individual patients, based on their map clusters, predict the degree of locomotor and just motor, even forelimb motor recovery on this ASIA impairment scale? And what we saw was that there's this very narrow window within which optimal blood pressure management yields better outcome. If you go too high or too low, patients have a lower probability of recovery. So this was pretty exciting because it's a clinical realization of something that we first found through machine learning on 20-year-old rat data. So I guess I would argue that through machine learning using old data, we're able to first do a successful replication and then ultimately a translation of these findings. And this paper came out this morning, led by Austin Chow in our group, that now takes this and pushes a broad framework for showing how you can commingle clinical expertise and machine learning to develop an expert augmented automated machine learning pipeline for looking at blood pressure potentially in real time down the road. So I guess the end of this story is that I think we've outlined sort of a machine learning almost cyborg way of doing this, where you have clinicians, data scientists, and machine learning tools all working together as sort of a collaborative unit. And in some cases, it can actually lead to what appears to be pretty remarkable recovery. When we published the paper in eLife, there was a press release, and it turns out that one of the patients in this cohort was this gentleman, Chris Barr, who was a surfer who came to our hospital after an accident early in the morning where he was a complete quadriplegic, and with very close blood pressure monitoring and spinal cord perfusion pressure monitoring, he left the hospital able to move and then subsequently engaged in a bunch of clinical trials, and he's now on his feet and is really quite functional. So the hope here is that this kind of thing isn't a fluke and that we can do more of this type of work in the future if we can come to bring machine learning and AI-ready FAIR data together with clinics. And I'll close there. I just need to point out where I work and the great number of people who are highly talented who I work with. And from there, I'll take some questions. Thank you.
Thank you, Adam. That was a really nice talk with excellent work put into it. So if everybody could please post their questions in the link that's in the chat. We have received a question that asks, have you used these machine learning tools for longitudinal data?
The short answer is yes, we have done some of that kind of work. So there are sort of variants of these types of tools that can look at clustering of trajectories over time. And there have been a few papers from our team and more on the way that are looking at this.
And another question is, how did you approach the standardization or the different variables to all be in the same multi-dimensional space?
Yes, that's a really good question. So there are different views for how one should do this. One view that I think has been championed by NINDS is to develop common data elements where everybody collects data, hopefully in the same way. I tend to think that that's never going to be complete CDEs are very helpful, but ultimately you need to have machine learning tools that can help you grapple with variability. And the nice thing about these nonlinear principal component analysis and similar tools is that they operate in a standardized space of correlation. You know, correlations are z-scores, like a Pearson R is a z-score. So they don't have to be the same variable. They don't even have to be on the same scale. They all get standardized from -1 to +1. And that helps you with the harmonization.
Thank you. I don't see any questions, but I had oh, we've got some more coming through. Could you speak a bit about how these open data platform tools have fostered data sharing internationally in the STI and TBI research fields, but also adhering to GDPR for EU data sets?
Absolutely, that's a really good question. So with the Open Data Commons for spinal cord injury and TBI, we've built it to be GDPR compliant, but of course we can't police this, you know, effectively. So we have requirements that if people are going to publish their data, it has to adhere to IRB sort of processes, and especially if it's clinical. But as long as you respect GDPR and make sure you don't share data that is inappropriate to be shared, it doesn't seem to be a problem. In addition, in the clinical studies, this becomes kind of a bigger problem. You're needing to bring together potentially HIPAA compliant data on a large scale. And for that, we need to have better federation infrastructure. So these are modern types of tools where data lives behind an encrypted firewall within your university, and you have a machine learning agent that can get in and get out. So it goes in, finds patterns, comes up to the cloud and reports to other sort of agents that have come together, and then you do this sort of federated machine learning. So it's becoming possible to do that.
And I think we have time for one last question. So a great talk, Adam, how the data sets from studies were curated before input into the database. And there's one more, let's, I think we'll have time for Dr. Agustin's.
Okay, so should I take both of these, HIPAA? Okay, so for data to be placed in the database, the Open Data Commons repositories, we have very minimal requirements technically. It needs to be a rectangular matrix where the variables are on a single row at the top. In addition, one of those variables needs to be the unique identifier for the subject. Those are the base requirements. Beyond that, in the TBI field, fortunately, because of HIPAA and others, we have emerging common data elements for preclinical and clinical TBI. So we have the ability to enforce those common data elements within the system. So people can upload their data, they name their variable whatever they want, but they can also say this variable is this other common data element. So they can link the two. And then the last question. Okay, NLP. Natural language processing is coming along. You know, there are some pretty advanced tools now that allow us to make use of even unstructured text data. I'll leave it there.
Thank you. So I'll turn it to you, Susan.
Thank you so much, Dr. Ferguson and Dr. Awad. This has been a fantastic and truly interesting seminar. I do appreciate watching the digitization of old records and data. Quite a heroic effort, and certainly paying off. And congratulations on your paper today. Thank you everyone who's attended. I hope that you will join us on May 13th. As a reminder, it's Dr. Karpathy Kramer. She'll be talking about her work to leverage data from multiple institutes and multiple cohort studies for supporting data discovery and data reuse. And she'll also talk about her educational programs at the Martonis Center for Biomedical Engineering at Mass General Hospital. So thanks again, and join us on May 13th.
Thank you, guys.
Thanks.
Thank you. Great job.